Gene expression and genetic switches. What is gene expression? The process by which a gene's information is converted into structures and functions of a cell is called as gene expression. Now, what is combination between the gene expression and genetic switches? There are certain genetic switches which are present to turn on or to turn off the gene expression. One of the example is the tryptophan repressor. Tryptophan repressor is a simple example that is a turn on and turn off switch of the gene expressions. What is repressor? Repressor is actually protein binds to DNA to prevent the transcription of adjacent gene. While on the other hand, what is activator? That is also a protein that binds to DNA and promotes the transcription of the adjacent gene. So in case of genetic switches, there can be repressor proteins and there can be activator proteins. Repressor proteins, which when attached to DNA, it prevents the transcription of the genes, and activators are those proteins when they bind to DNA, they promote the transcription of the adjacent genes. Gene expression and genetic switches, as the example is of tryptophan repressor, that is a simple turn on and turn off switch. Here we can see that inactive repressor. When there is inactive repressor, the genes are on. In other words, RNA polymerase can bind to the promoter sequence and the genes, RNA polymerase can produce the messenger RNA. While on the other hand, when there is a tryptophan and tryptophan binds with and the repressor becomes active. When a repressor becomes active, so the genes, they become off. So this is difference that what is inactive repressor? Inactive repressor, when there is inactive repressor in case of tryptophan, the genes are on. When there is an active repressor, a repressor will bind to the promoter sequence, to the operator promoter or operator sequence, then the genes, they become off. So this is a phenomena that what is to switch on and what is to switch off. So genes, they are on and genes, they are off with the help of repressors or activators, which are called as genetic switches. Here we can see that how the inactive repressor, this is the inactive repressor. When inactive repressor do not bind with the DNA, then the genes, they are on. When there is an active repressor in the presence of tryptophan, and the active repressor, it will bind to the operator region of the DNA, then the genes, they become off. So here we can see that this is an example of inactive repressor and active repressor. So when there is inactive repressor, genes are on. When there is active repressor, here we can see that this is an active repressor, then genes, they are off. Another type of the genetic switches is in the form of catabolite activator protein that promotes the transcription of genes that enables E. coli to use alternative carbon source when glucose is not available. So this is the diagram that how the cap protein binds with the DNA. There are some complicated genetic switches, and complicated genetic switches combine positive and negative control. They can on one hand perform the positive control, while on the other hand they can perform the negative control. Lack operon under the control of transcription activator and transcription repressor is the example of the one of the complicated genetic switches. Here we can see the diagram of lack operon that is under the control of transcriptional activator and transcriptional repressor. Here is the example. This is the gene, and here we can see that if there is glucose, means glucose present, lactose present, then the operon, they are off. If glucose is present and there is no lactose, repressor will bind and operon still they are off. While in this diagram, if there is no glucose, no lactose, then operons are off. While here we can see that if there is no glucose but there is lactose, then operons, they are on. Operon is actually a group of genes which is present in case of bacteria. Regulation of transcription in case of eukaryotes is more complex. In case of eukaryotes, we see that there are exons and introns. This is the diagram of a eukaryotic gene, in which we can see that here are the gene regulatory proteins. They are present here and they are also present here. So gene regulatory proteins can also be present in the downstream sequences. So genes, they can be controlled from the upstream sequences and they can be controlled from the downstream sequences. This is the gene control region that starts from the 5 UTR regions, that is 5 end of the genes as well as towards the 3 end of the genes. So this is all gene control region in which there can be exons and introns. And here is the poly A addition site. There are different transcription factors that control the expression of the genes. Like here we can see that there is GATA1, then EKLF, then CP1, then NF1. So these are all present at different positions starting from -30 up to -20. While on the other hand, here we can see that this is also the gene control region, where we can see starting from 2000 base pair away from the poly A tail up to the 2400 nucleotides. There are also two regions or the proteins that bind to the sequence of the DNA that control the transcription. So if we conclude that what are genetic switches? Genetic switches actually are those proteins, they either perform to turn on the gene or to turn off the gene, that can be in the form of prokaryotic cells or the prokaryotic genes, and they can be in the form of eukaryotic genes. In case of eukaryotic genes, genetic switches, they are more complex as compared to the prokaryotes.